Hello everyone, welcome back to the class. So far we have discussed about the endoplasmic reticulum's function. First one was N-glycosylation, and next one is synthesis of membrane lipid. This is the image of membrane lipid synthesis, and we had gone through different steps involved in this membrane's lipid synthesis, which is occurring in the cytosol. So after this, after the synthesis of the membrane lipid, what is the next step? We'll deal that today. Okay, so the first membrane lipid is synthesized in the cytosol, and then this phospholipid will be transferred inside the membrane, inside the bilayer, inside the inner leaflet of the bilayer. So what helps to transfer this phospholipid inside? That is scramblase. Scramblase are transport protein which helps in transport or movement of phospholipid from outer to inner and inner to outer leaflet of the bilayer. It performs both in both direction. It transfers in both direction, that is from outer to inner and inner to outer. This is its function. The other one is flippase and floppases. Flippases are the transport proteins which transfers or moves the phospholipid from outer leaflet to inner leaflet. Floppases are doing it oppositely. It transfers the membrane lipids or membrane proteins from inner leaflet to outer leaflet. Both flippase and floppases are ATP dependent membrane proteins, but this scramblase is very special as it does not involve any ATP usage. It is ATP independent. So scramblases is a membrane protein which transport protein which is ATP independent transport protein which transports the phospholipid from outer, in this case from outer cytosol to inner lumen. You can see in this image where phospholipid synthesis adds to cytosolic half of the bilayer. It causes the asymmetric growth of the bilayer. Scramblase catalyzes flipping of phospholipid molecule from the cytosolic to luminal leaflet. Symmetric growth of both halves of bilayer happens. Imagine this is membrane. Imagine this is membrane. The inner region which I am marking is the lumen. The outer one is cytosol. This is cytosol. From cytosol to inner lumen. So this is the direction where the transport or movement of phospholipid after synthesis is happening with the aid of scramblase. Mediates the transfer of lipid molecules from the cytosolic lipid bilayer to the non-cytosolic lipid bilayer to ensure that both monolayers remain equally populated with lipids. Transport of proteins from ER to mitochondria by phospholipid exchange proteins. These proteins are water-soluble exchange proteins, move phospholipids from the cytosolic phase of the ER to the cytosolic phase of the mitochondria and other organelles. What is the function of this transferring or movement of membrane proteins or membrane lipids? It detoxifies the xenobiotics. In SER, the smooth endoplasmic reticulum or hepatic cells, are major site of removal of xenobiotics. Then what is this xenobiotics? Xenobiotics are synthetic compounds which are not found in nature. You can see here cosmetic, diet, drugs, environmental pollutants are all xenobiotics. When it is exposed, the hepatic cells become what? Toxic. It becomes toxic. It forms some reaction inside the cell. So the cytochrome P450 catalyzes this process. Monooxygenases, which are heme containing enzymes that catalyze the incorporation of a single atom of molecular oxygen into the substrate with the concomitant reduction of the other atom to water. In the hepatic cell, the cytochrome P450 are membrane bound and are important for detoxification of xenobiotics and catalyzes reaction. Here in this reaction, RH refers to the xenobiotics. The phospholipid lipophilic xenobiotics are thereby converted to more water soluble form, which aids in their excretion by kidneys. This hepatic cells helps in detoxification. Whichever xenobiotics has been exposed, which are mutagenic, carcinogenic, which are toxic to the cells, that are what reacted with this molecular oxygen, with the molecular oxygen, and with the help of transferases it undergoes different steps, so that at last it becomes more water soluble form and it aids in the excretion through kidney. This is the main function. So next topic is endomembrane system, which is quite interesting from here. Till now you have learnt the ribosome structure, its function, endoplasmic reticulum and its function. So protein is, as of now, it has come here and it is about to exit from the endoplasmic reticulum to Golgi. So this endomembrane system involves a set of membranes that form a single functional and developmental unit, which helps in transport of different types of proteins across them. See, these are in line. The nucleus, endoplasmic reticulum, Golgi apparatus, lysosomes, secretory vesicles, etc. are involved in the endomembrane system, which are either being connected together directly or exchanging material through vesicular transport. See, these vesicles helps in transport. Composed of membrane bound structures that are suspended in cytoplasm of the eukaryotic cell. These membranes divide the cell into functional and structural compartments. The membrane bound structures of the endomembrane system, which are the organelles present in nuclear envelope, endoplasmic reticulum, Golgi complex, lysosome, vacuoles, transport vesicles, are all involved in transport of proteins. So this all organelles comprises of unit called endomembrane system. Let us look into it. First process is transport of proteins across the ER membrane. As of now, just now you learnt that ER membrane will transfer some of the proteins. It also produces or it also synthesizes proteins. Some of the modification of proteins happens in the ER. This modified protein, where it will go across the ER membrane and at last it goes to the Golgi, and at last the destination is the cell surface. Let us know one by one. Proteins synthesized by membrane bound ribosomes include soluble proteins and membrane proteins. So membrane bound ribosomes, what do I mean this term membrane bound ribosome? That is ribosome associated with the endoplasmic reticulum. Which type of proteins do that produce? Soluble proteins and membrane proteins. These proteins reside in the ER. Proteins destined to be secreted move through the secretory pathway. From rough endoplasmic reticulum, it goes to endoplasmic reticulum's lumen, then Golgi transport vesicles, then it goes to Golgi cisternae, then there is two pathways, secretory pathway and transport pathway. What protein will get into any one of this pathway, and at last it moves to the cell surface. Secretory proteins are targeted to ER by a signal sequence. In the first class only I said, for protein transport or the protein to be targeted, it should have a what signal sequence as its tail. In its tail there is a particular sequence, that is signal sequence. Secretory proteins which are targeted to ER by a signal sequence. What is this signal sequence? A short sequence of 15 to 35 amino acids that contain a sequence of at least 6 non-polar amino acids at the end terminus of the growing polypeptide chain. So this is a signal sequence. These signal sequence are cleaved from the polypeptide chain during its transfer into the ER lumen. Once it is transferred, the protein the polypeptide chain will be detached from the signal sequence. How it happens? There is a signal recognition particle which is having two binding sites. Consider signal recognition particle. It is having two sites. One side binds to the signal sequence, second will binds to the ribosome. Consider this as a ribosome which is producing the polypeptide chain. So one end binds to the signal sequence. Consider this as a signal sequence. The first end, the signal recognition particle is having two ends, right? One end will bind to this signal sequence. The other end binds to, it is already bound here. Second end will bind to the ribosome. Then this forms a complex. This forms what? Complex. That is SRP, nascent polypeptide plus ribosome. There is a polypeptide chain here. There is a ribosome here. There is SRP here. That forms a what? Complex of SRP nascent. What is this? What is the fate of this complex? This complex, consider, I'll write it in a short way, SRP plus polypeptide chain plus ribosome. It is what? A complex, right? All this will bind to the alpha subunit of what? Alpha subunit of SRP receptor. SRP receptor. SRP polypeptide nascent polypeptide ribosome complex will bind to what? Alpha subunit of SRP receptor. Where this receptor is present? It is present in ER membrane. ER membrane. So let us see now. So signal recognition particle has two binding sites. One will bind to the polypeptide chain, other will bind to the ribosome. This forms a complex. This complex binds to the alpha subunit of the SRP receptor. So I'll write here complex plus alpha subunit of SRP receptor. So in this SRP is there, right? SRP directs both binding of the ribosome to the ER membrane and insertion of the nascent polypeptide into the transmembrane channel. In previous slide we saw that the membrane bound ribosome will synthesize soluble proteins and membrane proteins, right? Where should these membrane proteins be targeted? Obviously to the membrane, right? So the SRP will direct both binding of the ribosome to the ER membrane, and also it helps in insertion of this nascent polypeptide into the transmembrane channel. This SRP here present, no, SRP and alpha subunit of SRP receptor, receptor, I'll write it once again, complex, what? Complex plus SRP receptor. Complex is having what? SRP. It is reacting or it is interacting with the SRP receptor. So after both of them will react, it directs toward the SRP ribosome complex to the translocone. What is translocone now? It is a gated translocation channel. Once the SRP and SRP receptor tries to interact with each other, they direct the SRP ribosome complex to the translocone, which is a gated translocation channel. N terminal of the polypeptide chain enters the ER lumen through the translocone. Consider this, I'll write it in a different color. Consider this is ER surface. Consider this is a ER surface. There is a translocone channel. Here, the yellow part. Okay. After this SRP and SRP receptor tries to interact, they direct the complex. I'll write it in a different color. This is a complex. The green one is SRP receptor. What does it contain? It contains SRP, nascent polypeptide, ribosome, SRP receptor, alpha subunit of the SRP receptor, right? It will be directed to the translocone. Especially the N terminal. What? The N terminal of the polypeptide chain enters into the ER lumen through this translocone. So consider there is a polypeptide here, right? The N terminal will be directed to the ER lumen through this translocone. The yellow color is translocone. Okay. So what had happened here? Complex of SRP, nascent polypeptide plus ribosome will forms a complex. Then this complex binds to the alpha subunit of the SRP receptor. This SRP directs both binding of the ribosome to the ER membrane and insertion of nascent polypeptide into the transmembrane channel. Then what happens? Interaction of SRP and SRP receptor then directs the SRP ribosome complex to the translocone, which is a gated channel. N terminal of polypeptide chain enters into the ER lumen through this translocone. What happens next when it is enters? Then it forms the hydrolysis of GTP of SRP and SRP receptor happens. Hydrolysis of what? GTP of SRP and SRP receptor. Once the hydrolysis of GTP of SRP and SRP receptor happens, the interaction between SRP and SRP receptor will close or ends. Both SRP and SRP receptor have a GTP binding domain and GTPase activity. When GTP bound to both the P54 subunit of SRP and the alpha subunit of SRP receptor, it leads to dissociation of SRP and SRP receptor. Polypeptide chain elongates. Here you can see small ribosomal unit, large ribosomal unit. Ribosome binds mRNA. It forms mRNA. Translation begins. polypeptide chain, it starts to translate. There is a signal sequence. SRP binds signal and ribosome, arresting polypeptide translation. Then there is a pause here. Unit binds to the SRP receptor on the ER membrane. Then SRP is released by hydrolysis of the GTP. Then the translocone, here it is that the translocone is present, where what the N terminal of polypeptide will be entering into the ER lumen. After this, signal sequence is cleaved. The signal sequence you can see here, signal sequence, N-terminal signal sequence will be what? Cleaved with the help of signal peptidase. The function of that signal peptidase is signal sequence is over, no? As the protein is directed to the ER lumen itself, then there is no function of signal sequence anymore. Signal sequence is cleaved by the enzyme signal peptidase. Let us see now. See, it is cleaved by the signal peptidase associated with the translocone protease, which recognizes the sequence on the C-terminal side of the hydrophobic core of the signal sequence and cleaves the chain and is rapidly degraded. C63 complex and membrane of HSP70 family, that is binding immunoglobulin protein, are responsible for unidirectional translocation across the ER membrane. So this C63 complex will help in unidirectional translocation of ER membrane. BIP has a peptide binding domain and an ATPase domain which binds and stabilizes unfolded proteins. The ATP driven cycle of BIP binding and pulls the protein into the lumen. So the protein is anyhow, it should be landed to the ER lumen. So it which is helping for it, C63 complex and membrane of HSP70 family, helping in unidirectional translocation of that protein. So till now what we had discussed about endomembrane system and transport of protein from cytosol to the ER lumen. So in next class we will be learning about transport from the ER back to cytosol. So from cytosol we had sent the protein to the ER lumen. So in the next class we will be doing it reverse. It will be transferring protein from ER back to the cytosol. Why back to the cytosol? We'll discuss it later. Thank you.